I think I haven't seen many more joining new. Well, a few people are still joining, but I think 5 minutes is good enough time to give everybody to populating the meeting. And welcome to everybody. Good morning. My name is Beppe Battaglia. I'm the group leader of the Molecular Bionics group, and it's my great pleasure today to chair this seminar, which will be delivered by our new addition. Well, not so new now, a few months already in, well, close in that joined in our group this January. Daniel González Carter, or Dr. González, joined our group through quite prestigious junior leader Aisha programs, and is now working in our group, building up his own activity around blood-brain barriers and getting across and understanding endothelial biology. Daniel is actually graduated from Nottingham University in the UK, and he then moved on into the PhD at Imperial College, where he also did a postdoc. After that, he moved all the way to Japan, where he joined a group of Professor Kataoka at the University of Tokyo, where he stayed there for a few years. And then finally, last year, he was successful in securing, well, 2 years ago, was secured his fellowship and started this January working with us. And since then, he's been a great addition to the group. So I'm very pleased to share the work he's done before, and perhaps some new work is done with here in our labs. Daniel, without any further ado, I'll let you take the lead and tell us about your work.
Okay, well, thank you very much, Pepe. Thank you very much for that very nice introduction. And thank you, everybody, for coming to the seminar. And I've entitled this talk, Delivering Nanoparticles Specifically to the Brain by Generating Artificial Brain Targets. So just a little bit more introduction about myself. As Pepe said, I did my PhD at Imperial College in neuroscience, looking at neuroinflammation and microglial activity with a focus on Parkinson's disease, but we also looked at the biology of the blood-brain barrier. And after that, I did a postdoctoral position. I moved to the Department of Materials to look at the interaction of nanoparticles, mainly carbon nanotubes and silver and gold nanoparticles, with brain cells, in particularly microglia and blood-brain barriers, or brain endothelial cells. And after that, I moved to Japan with an early career scientist research grant from the Japanese Society for the Promotion of Science to work under Professor Kazunori Kataoka at the Innovation Center of Nanomedicine in affiliation with the University of Tokyo. And there I was focusing mainly on developing nanoparticles that are decorated with glucose to be able to deliver therapies against neurodegenerative diseases into the brain. But I also started to establish this new brain delivery strategy where we exploit the blood-brain barrier impermeability to selectively label the brain vasculature, which will be the main focus of this talk. After that, I moved here to Barcelona. I started working actually in March due to a little delay in the red tape. But I started in March this Lacacia Research Fellowship, where I will be looking to further develop this novel brain targeting strategy. So let's get into it. Why do we need brain targeting strategies? And the answer is because it's very difficult to get therapies from the blood into the brain. And this is because the brain vasculature is lined by this protective cellular layer made of specialized endothelial cells of the brain vasculature, which, because of very close interaction between astrocytes and pericytes, have a very high impermeability. Both paracellular impermeability, because of the presence of tight junction complexes, which are interendothelial proteins, which bind neighboring endothelial cells very tightly. So there is very little leakage in between the cells. And they also have very high transcellular permeability, because brain endothelial cells have a very low rate of endocytosis. so very little gets through the endothelial cells. Despite this high impermeability, there are innate mechanisms which the brain endothelial cells use to get nutrients and hormones and other proteins through the BBB, through the blood-brain barrier, and into the brain. For instance, there is carrier-mediated transport, where molecules are incorporated into endothelial cells by channel proteins and then exits into the brain parenchyma. Examples of these are the glucose transporter GLUT1 and also amino acid transporters such as SLC7A5. And there's also receptor-mediated transport, where proteins bind to receptors on the cell surface, are internalized through endocytosis, and are actively transported in vesicles through the endothelial cell, and finally are exocytosed. And right now, all the current brain targeting strategies try to exploit these transport mechanisms to get nanoparticles into the brain. So many different strategies have used glucose decoration of the nanoparticles to bind to GLUT1, transferrin or anti-transferrin receptor antibodies decoration to bind to transferrin receptor, as I say, amino acid functionalization to target solid carriers, or angiopep decoration to target LRP1, and thereby release the nanoparticles in the brain. And these strategies have had success, and some of them are being tested in the clinic right now, but all of them have inherent brain specificity limitations. And this is because all of these proteins, even though they are overexpressed at the brain vasculature, they are also significantly expressed in the peripheral vasculature, so in the vessels of the lung, liver, and heart, for example. Therefore, there is a very high rate of off-target nanoparticle uptake, meaning that even though you increase nanoparticle uptake in the brain, you also increase it in other organs. Therefore, this decreases the therapeutic efficiency and might increase the toxic side effects, which ultimately will limit clinical translation. And these are just two papers recently published to highlight these problems. And here in the Nature Communications paper, they complexated RNA with a peptide targeting transferrin receptor 1, and they do see a good increase in brain uptake, about 4-fold, but they also see a strong increase in lung uptake, about 10-fold. Here in Science Advances paper, they generated lipid nanoparticles decorated with neurotransmitters to exploit neurotransmitter carrier transport, and they again see a strong increase in brain uptake, but they also see a strong increase in liver and lung uptake. Therefore, these limitations need to be overcome. And to achieve that, we're trying to develop this new targeting strategy, where instead of using endogenous proteins, let's say, we instead generate the targets artificially, thereby having very specific brain uptake. So it should increase brain specificity of nanoparticle delivery. And we are using, actually perhaps counterintuitively, the impermeability of the blood-brain barrier itself to achieve this. So if you remember, I said that the impermeability of the blood-brain barrier is, at least in part, due to a very low rate of endocytosis into brain endothelial cells compared to peripheral endothelial cells. And this is because brain endothelial cells express this lipid incorporating protein, MFSD2A, which incorporates DHA into the cell membrane, and thereby prevents association of CAV1 with the cell membrane, and therefore reduces the rate of endocytic vesicle formation. This was very nicely demonstrated by the group of Chenghua Gu in Harvard University, where they show that if you knock down MFSD2A expression in mice, you see a much higher leakage of, in this case, they were using the tracer dextrin, 10 kilodalton dextrin. And they showed that this leakage is not due to a disruption of the architecture or of the tight junctions of the blood-brain barrier. So you can see that the architecture of the tight junctions is fairly similar between the wild type and the knockout mice. And the leakage of the tracer in between the tight junctions is similarly prevented in the wild type versus the knockout. What is different is that while in the wild type there is very little endocytic vesicle formation, in the knockout you can see that the tracer is taken up into the vesicles and transported across the endothelial cells to be released in the abdominal side. And also, they showed that using the BEN3 brain endothelial cell line, which at least in their hands does not express MFSD2A, that this cell line has very high rate of endocytic vesicle formation. But if you rescue MFSD2A expression, this rate of endocytosis is decreased, meaning that the blood-brain barrier phenotype of these cells is rescued. And here I would just like to give a cautionary note to anybody modeling the blood-brain barrier with this very popular cell line, that make sure you have a good expression of MFSD2A, because otherwise your endocytic rates will be modeling more peripheral endothelial cells as opposed to brain endothelial cells. But so using this aspect of the blood-brain barrier, we are trying to generate brain-specific targets through this strategy, where we basically firstly, we firstly attach a molecular label to the endothelial cells of the whole body. Then, because of the lower rate of endocytosis in brain endothelial cells compared to the peripheral endothelial cells, this label is removed more quickly from the periphery than from the brain, meaning that after a certain period of time, there is only selective labeling of the brain vasculature and thereby creation of brain-specific targets. Then, by generating nanoparticles functionalized with a linker which efficiently recognizes this target, we should be able to have no interaction with periphery and only specific interaction with the brain vasculature and thereby brain target. And so last year we published a paper on PNAS where we showed that this works at least in principle. And we used biotin to label the endothelial cells and abidin as the linker on the nanoparticles. We used this system for two reasons. Firstly, biotin and abidin bind very strongly. Therefore, it will allow us to have efficient label recognition. But also, abidin binds 4 biotins per protein. Therefore, it would be able to kickstart endocytosis through protein clustering. However, I have to stress that this is only, we chose only this for convenience sake. It is not intrinsic to the strategy, and therefore we can actually think of using other labels and other linkers to generate the artificial targets. So just a little bit about the nanoparticle synthesis. We used pegylated polyionic nanoparticles. We created them by complexating polyionic polymers with polycationic polymers and generating azide decorated nanoparticles, which have Cy5 fluorescence inside to be able to quantify them and image them. And then we used a FITC conjugated abidin DBCO protein to attach abidin onto the azide functional groups. We generate nanoparticles of about 50 nanometers with a PDI of 0.149, with so a monodispersed population and a fairly neutral zeta potential. And I won't go into the details, but I'll just say that through fluorescence correlation spectroscopy and analytical ultracentrifugation, we calculate that more or less we're having between 3 or 4 avidin proteins per nanoparticle. Therefore, we wanted to see if these avidin nanoparticles could actually be endocytosed into brain endothelial cells following biotin target generation on the cell surface. And to that end, we extracted brain endothelial cells from rats, and we biotinylated their cell surface by using this biotin compound, which by the use of NHS binds to primary amines on cell surface proteins. So we can see that the avidin nanomicelles here in blue have a much stronger incorporation compared to blank nanomicelles, meaning that the avidin is being able to use the biotin targets to internalize the nanoparticles into the endothelial cells. And importantly, this internalization is decreased if we incubate the cells at 4 degrees, meaning that it is an active endocytic process. We corroborated this by confocal microscopy. And you can see that it is only when the when the cells have been biotin labeled and the nanomicelles have avidin that there is strong Sci5 fluorescence in the cells. At 4 degrees, you can see that there is still strong Sci5 fluorescence, but I hope you can appreciate that it is only restricted to the cell surface, meaning that the avidin nanoparticles can bind the biotin targets, but because of the lack of energy, let's say, they're not able to be endocytosed. Then we wanted to see if we could actually use the lower rate of endocytosis of brain endothelial cells to specifically retain the biotin targets on the surface of brain endothelial cells. And so to that end, we again biotinylated the cell surface of either brain endothelial cells or peripheral endothelial cells. And then we waited 0, 1, 2, or 8 hours to allow for the biotin target to be removed by endocytosis. And then we treated with the avidin nanomicelles. And therefore, you can see that as the time goes by, the ability of the avidin nanoparticles to be internalized into the peripheral endothelial cells steadily decreases, while it is pretty much maintained on brain endothelial cells. And this is corroborated by confocal microscopy, where with no biotin target generation, there is very little nanoparticle uptake in both cell types. But immediately after biotin target generation, there is a strong increase of uptake in both cell types. But at 8-hour time interval, this is only the case for brain endothelial cells, meaning that after 8 hours, there is very strong removal of the biotin targets in the peripheral endothelial cells, but not from the brain endothelial cells. Then we wanted to see whether this could be retained in vivo. And to that end, we used a biotinylated anti-PCAM1 antibody. PCAM1 is platelet endothelial cell adhesion molecule, which is an interendothelial protein expressed in the whole, in the vasculature of the whole body, but it is more strongly expressed in the periphery versus the brain. And importantly, it has been shown to be associated with CAF1. Therefore, it was a good protein target to try to see if we could exploit the lower rate of endocytosis of brain endothelial cells in vivo. So first, we wanted to see the binding of the antibody to the vasculature wall. And to that end, we used a slightly different antibody. In this case, it is a fluorescent anti-PCAM1 antibody. And we injected it intravenously to mice. And then we imaged the antibody distribution in the vasculature by in vivo confocal microscopy. We were looking at the ear vasculature. And so you can see at 10 seconds after antibody injection, it is present all around the artery, so in the lumen, and also, I guess, in the cell wall. But steadily, as time goes by, the lumen signal steadily decreases, and after 10 minutes, you can see that it is lining the vasculature very nicely And after 1.5 hours, the only signal that we can detect is at the brain vasculature, meaning that the antibody has labeled the vasculature of the walls of the vasculature and is no longer circulating in the blood. Then we return to the biotin-related anti-PKM1 antibody, and we again injected it into mice intravenously. And then after a time interval of 15 minutes, 2 hours, 4 hours, or 8 hours, we inject the avidin nanomycin cells. And then after 16 hours, we perfuse the animals and we quantify nanoparticle uptake into the different organs. So you can see that at the short time interval, there is strongest nanoparticle uptake into the lung, or nanoparticle uptake increase, I should say, into the lung, but there's also into the brain, heart, and the pancreas. However, as the time interval between antibody injection and avidin nanoparticle injection increases, the targeting ability of the nanoparticles into the lung steadily decreases, as well as to the heart and the pancreas. But it is fairly maintained at a constant rate to the brain, meaning that after an 8-hour time interval, the only increased uptake that we see is in the brain, meaning that the targets are only present in the brain vasculature at this time, allowing for specific nanoparticle uptake increase in the brain. And here you can see the distribution of the nanoparticles if we inject only with PBS or if we inject with the biotin-related anti-PKM1. You see that by IBEC, so this is freshly excised organs, we can see a very strong increase in nanoparticle fluorescent signal. And also in fixed tissue, we can see that the brain tissue has much more nanoparticle distribution if we use the biotin-related anti-PKM1. So all of this data allow us to say that yes, the principle of using the lower endocytosis of brain endothelial cells to generate artificial targets works. The proof of principle is there. However, as the strategy stands, there are several shortcomings and points which could be improved, which is what will form the basis of my Caixa fellowship. And two shortcomings which I will focus on right now is, firstly, we have shown that this works if we target PKM1, but targeting PKM1 is not ideal because, as I said, PKM1 is an interendothelial protein. So whatever targets we might generate will only be at the interface of endothelial cells. Therefore, we will have limited target generation. And it could be that there are other proteins in the vasculature which have a much better endocytosis or endocytic rate profile, meaning that there might be other proteins which are retained at the brain vasculature more specifically. Therefore, we are going to use phage display technology to find peptides that are retained on brain endothelial cells with a wider cell surface distribution, allowing us to have much higher rate of target generation, but also a more pronounced endocytic rate difference. And therefore, that will allow us to have a very quick generation of the specific brain targets, which might be in the lower time frame than 8 hours. The second limitation of the strategy right now is that we are I'm gonna echo from myself. Okay. Yeah, we are only transiently generating the targets. I will explain a little bit further about this later on, but I'll just say that this limits the strategy to either single dosing or very limited dosing, which is inefficient for the treatment of chronic diseases like Alzheimer's disease. And to overcome this, we are developing an mRNA delivery platform to be able to continuously regenerate the targets, which will allow us to have multiple dosings. So, we'll talk a little bit about what the phage display is. Here we are using phages, which display different peptides or a single peptide on their surface. And the library of the phage allows it to have about 10 to the 9 unique peptide sequences. So you have a very large population of peptides which you can select from. And by doing biopanning, you can stepwise lower the range of these of the peptide variety until you, and by enrichment, until you reach a number of peptides which is suitable for sequencing and have the right characteristics which you are looking for. We will look at phages which not specifically bind only to the brain vasculature. This has been done before, but those phages that bind to the vasculature of the lung, liver, and the brain, but are specifically retained on the brain endothelial cells by quickly being removed by endocytosis from the liver and the lung. And after a certain period of time, we collect the phages that are only displayed on the cell surface, and we should be able to synthesize the peptides from them and hopefully have candidates which will allow us, as I say, to have a high number of cell surface target generation in a short time interval. Okay, I don't know why this is a little bit moved. All of the organs obviously should be in the brain, in the body, but it should allow us to have peptides which we should be able to inject into the patient. They will bind to the vasculature of the organs, but after a certain period of time will only be present in the brain vasculature. And more interestingly, we could actually find different combinations of peptides which would create a target pattern on the brain vasculature, which we can exploit to have very specific nanoparticle delivery through a superselectivity strategy which the group of IBEC is generating. So hopefully we should be able to have very specific nanoparticle delivery to the brain by generating the artificial brain targets. Right now, I don't have any interesting data regarding the phage display. So this is all I will say about this right now. But in the last 10 minutes or so, I want to talk about about how we are overcoming the transient generation of the targets by delivering mRNA. So, why are the targets being generated transiently? And it's because as the targets are displayed and the nanoparticle binds to the targets, its endocytosis will also endocytose the targets. Therefore, they are depleted, meaning that at a second or subsequent dosing, the nanoparticle will not be able to have any targets onto which to bind. And this is nicely illustrated in this graph here, where we biotinylated the cell surface of the endothelial cells, brain endothelial cells, and then we treated either with blank nanomicelles or with avidin decorated nanomicelle. And you can see that at the early time points, there is very strong increase in nanoparticle uptake with the avidin functionalization. But as the time goes by, the uptake increase steadily decreases, meaning that after a time point of about 2 hours, the uptake rate is very similar between the blank nanomicelles and the avidin nanomicelles, presumably due to the depletion of the biotin targets. Therefore, this target depletion, as I say, will limit the dosing, which means that we would need to regenerate the whole targets all over again, which would be time consuming and might also induce peripheral toxicity, because if we're asking the peripheral vasculature to continuously remove these peptides, these targets, we might end up having adverse effects, which will clinically be restrictive. Therefore, we want to regenerate the targets to allow for multiple dosing. And for that, we are using, or we are developing a strategy where not only do we deliver therapy-loaded nanoparticles, but also in conjunction deliver nanoparticles which are loaded with mRNA. And therefore, as they are endocytosed and release the mRNA, this mRNA would be encoding a labeled protein, which would be transported to the cell surface and display the label on the cell surface, and thereby regenerating the targets, allowing a second dose of therapy-loaded nanoparticles to bind to the target, be endocytosed and delivered into the brain, but also the mRNA nanoparticles. And this would, again, regenerate the labeled protein for regeneration of cell surface targets, and we could go on, I guess, indefinitely dosing the therapy. To achieve this, we are right now using a 2-protein system. The first protein being a biotin ligase, which activates endogenous biotin by attaching AMP onto it. And the second protein is a transmembrane protein. In this case, we are using the transmembrane domain of the platelet-derived growth factor receptor beta. And this transmembrane is fused to an acceptor peptide, so this sequence, which very specifically accepts the activated biotin from biotin ligase and attaches it covalently. Therefore, we end up with a transmembrane protein having on its end a covalently bound biotin. Therefore, by expressing these proteins within the cell, the endogenous biotin, free biotin in the cell, should be activated, bound to the acceptor peptide fused to the transmembrane protein, and as it is translocated to the cell surface, will display the biotin on the cell surface, allowing the nanoparticles functionalized with avidin to bind. So firstly, we wanted to see whether cells had enough free intracellular biotin to allow this to work. And for this, we used lung epithelial cancer cells, the A546 cell line, and we transfected them with either a plasmid encoding the transmembrane protein, a plasmid encoding the biotin ligase, or both in combination. And then after 24 hours, we treated, in this case, with free avidin FITC, or with free Fab prime to antibody fragment, also conjugated with FITC, simply as a control protein. And then we quantify how much protein binds to the cell surface. And so you can see that in comparison to control cells, so without any DNA transfection, the cells transfected with single proteins, so either the transmembrane protein or the biotin ligase protein, have very little and actually non-significant increase in both avidin and Fab prime to binding. But it is only when we give both proteins in combination that the binding of avidin to the cell surface increases significantly. And importantly, this does not occur for Fab prime 2, meaning that by giving the DNA in combination, we're able to generate enough cell surface targets to allow biotin targets to allow avidin to bind. Then we wanted to see if this could still work if we use mRNA instead. So we synthesized mRNA from these two from these two plasmids. Then we conjugated it with polycationic pegylated polymers to allow us to generate polyplexes to protect the mRNA from degradation. This is just a DLS characterization of the polyplexes, and we can see that in comparison to the DLS distribution of free mRNA, we are reducing the aggregation of free mRNA, and we end up with more or less a single peak polyplex DLS distribution. And we characterize the size, and we have about polyplexes of about 60 nanometers with a more or less monodispersed distribution of 0.2 and 0.23 for the transmembrane polyplexes. Okay, then we wanted to see if these polyplexes could indeed deliver the mRNA for generating the cell surface targets. And so again, we used the lung epithelial cancer cells, and we treated them with a combination of naked mRNA of both biotin ligase and the transmembrane protein, or polyplex mRNA, again in combination, or polyplexes encoding or incorporating only biotin ligase mRNA to see the interaction between the two proteins. You can see that in comparison to, well, to no mRNA, the naked mRNA has, sorry, I should say that then 15 hours later, we treat with avidin FITC and then quantify the amount of avidin binding to the cell surface. You can see that the naked mRNA condition has a slight increase in avidin binding, but it is not significant. But this is much more strongly, the avidin binds much more strongly if we use the polyplexes, meaning that the mRNA is being protected and delivered to the cells. Here we see confocal images of the avidin binding to the cells. For some reason, we are also having increased avidin binding when we only use the biotin ligase mRNA. And this could be either due to unspecific effect of the polyplex itself, or perhaps our biotin ligase is not as specific for the acceptor peptide of the transmembrane as we thought. So maybe it is actually attaching the biotin onto other cell surface proteins. We are not entirely sure about that at the moment. But what is encouraging is that this increase in avidin binding is specific for avidin. So if we treat instead with albumin with the same conditions, so either the biotin ligase mRNA alone, the transmembrane mRNA alone, or both in combination, we see avidin binding, but no albumin binding. Therefore, we are quietly confident that the biotin targets are being generated on the cell surface. And also importantly, if we increase the availability of free biotin within the cell by treating the cells with exogenous biotin before the mRNA polyplex treatment, we see an increase in avidin binding, specifically when we give the mRNAs and not when we don't give the mRNAs, meaning that the biotin supplement is not simply binding to the cell surface, but is actually being internalized, and then it is being displayed on the cell surface by the biotin ligase attaching it to the transmembrane protein. So, in conclusion for this mRNA delivery part, we are able to express biotin ligase and the engineered transmembrane protein to allow endogenous free biotin to be displayed on the cell surface to serve as targets for avidin binding, and we are successfully complexating it into polyplexes for delivery into cancer epithelial cells to generate the cell surface targets. Obviously, we need to corroborate this in primary brain endothelial cells to see if we can actually deliver the proteins and express them, and also to see whether the primary brain endothelial cells have enough intracellular free biotin available. And we need to functionalize the polyplexes with avidin to allow them to bind to the initial cell surface targets, which we would generate through the peptides identified through phage display assays. But more further along, if we are able to establish this mRNA delivery platform specifically to the brain, we can start to think about strategies which don't necessarily use the biotin-avidin interaction. So we can actually start to express engineered proteins on brain endothelial cells and only on brain endothelial cells, which will serve for selective recognition of functionalized nanoparticles. Okay, so to wrap up, what we hope to have at the end of this fellowship, or at least the work that will allow for further development at the end of this fellowship, is to have identified peptides which will allow us to have very efficient initial target generation on the cell surface of only brain endothelial cells, and then have a mRNA delivery platform which will allow us to have continual target regeneration from within the cell. And therefore, this strategy should allow us to have increased nanoparticle delivery efficiency, which would ultimately, in the clinic, if we are successful enough to reach those stages, would reduce therapeutic dosing and therefore would decrease treatment costs, but also would decrease peripheral side effects, which would allow us to increase the clinical translation of nanoparticle-based therapies. But I think also very importantly, what generating artificial cell surface targets would allow us to do is to avoid using up essential blood-brain barrier transporters like transferrin 1 or GLUT1, because they are overexpressed on the brain vasculature for very important reasons, right? To be able to maintain close control of brain homeostasis. Therefore, if we start to use them for our delivery strategies, we might end up down the line, especially if we use them chronically for treatment of, let's say, Alzheimer's disease, we might end up disrupting the brain homeostasis and therefore generate detrimental side effects, which would limit their clinical use. And just to finish up, I would like to thank everybody that has made this work possible. Obviously, everybody at the Innovation Center of Nanomedicine in Japan, but in particular, Professor Kazunori Kataoka for his brilliant guidance in the application of polymers for medical in the medical field, but also Dr. Anjana Yulu Dirsana for being very generous in providing me the polymers to do mainly the mRNA delivery. And then here at IBEC, at the Molecular Bionics Group, everybody that is working with me, but in particular Professor Beppe Battagne for supporting the fellowship, and also Julia Porro, visiting master student, who helped a lot with the mRNA work. I will also need to thank the funders that made this work possible, so JSPS in Japan, and the La Caixa Foundation for the fellowship here in Spain. And well, lastly, you for listening, and I'll be very glad to take any questions and try to give you answers.